And my file corrupted, so we are going to pretend that I didn't just record this. So, blood glucose is a very important controlling mechanism for the body. We need sugar in our blood to do stuff, right? Like our brain needs sugar in order to think, our muscles need sugar in order to contract, all of our organs need sugar in order to function. And so the amount of sugar that we have in our blood is a very important number for our body to control. If we have too much sugar, our body can't function. If we have too little sugar, our body can't function. So after you eat a meal that is rich in carbohydrates, rich in sugars, we're going to see insulin, a hormone, do its job to get rid of that excess blood sugar and store it somewhere. We store it in our liver, and we store it in our skeletal muscles, and we also store it in our fat, in our adipose tissue. When blood glucose is low, we're going to see processes controlled by the hormone glucagon in order to increase blood sugar. Those two processes are glycogenolysis and gluconeogenesis. Now, when you eat a carbohydrate-rich meal, your blood sugar goes up because the sugar goes from your gut, your digestive tract, into your bloodstream. You have increased blood sugar. This signals the pancreas, the pancreatic islet cells, to increase the amount of insulin in the blood and decrease the amount of glucagon being released. Increased insulin secretion leads to the formation of glycogen formation in the liver, and the formation of fat in adipose tissue. This happens because insulin tells the cells to absorb glucose and use it for glycogen formation. By absorbing the glucose out of the blood, we reduce the blood sugar and get back to a nice normal amount. Now, if you're fasting or exercising, then you're using up sugar faster than you're consuming it. You're reducing your blood sugar faster than you're eating meals in order to replenish your blood sugar. Then you're going to see your pancreas respond by releasing glucagon. Glucagon is a hormone that tells the body, I don't have enough blood sugar, this is an emergency, we need to increase the amount of sugar in the blood. The way that happens is by two processes. One is the breakdown of glycogen, which is glycogenolysis. We break down glycogen to release glucose. And the other process is called gluconeogenesis. Gluco, sugar, neo, the production of new sugar. So the liver can convert other molecules into glucose if we're fasting and we don't have enough blood sugar. Fasting, of course, means not eating. Now, that energy that comes from food can be utilized via cellular respiration. So if I eat food, I can convert it into glucose and then use cellular respiration to turn my glucose into ATP. But the food also provides organic carbon and organic compounds that are just useful in general for the synthesis of necessary biomolecules. Now, a topic I want to spend a little bit of time on here is basal metabolic rate. Basal metabolic rate means basal is like low baseline level, so the minimum amount of energy that your body consumes under defined resting conditions. So if I measured it as, say, while I'm laying down on a bed, if while I'm laying down on a bed, you might not think that I'm burning calories or using energy, but to breathe in and out, there are muscles involved in breathing, so those muscles take energy. To think, the brain uses sugar, that sugar is energy, so just laying down in bed and doing nothing but breathing and thinking is enough to burn calories. Now, if you measure it with defined conditions over a period of time, you come up with your basal metabolic rate. And your basal metabolic rate is useful for a couple of different weight loss applications. There's an interesting diet that looks at the amount of calories you consume and the amount of calories that you burn. And the idea behind it is, hey, if your basal metabolic rate is, say, 1,000 calories a day, and you eat exactly 1,000 calories a day, then in theory, your body weight shouldn't be changing as long as you're on that diet. And if you eat less than your basal metabolic rate, that means you'll burn calories every day, and as a result, you'll have a net loss in weight. It's an interesting theory. Now, if you eat too much food, if you eat more than your basal metabolic rate, then the excess food is stored as glycogen, and once your body runs out of space to store glycogen, which is pretty quick, then it starts accumulating it as fatty tissue or adipose tissue. Now, in order to control how hungry we are, there are a couple of hormones that serve key functions. I'm only going to focus on one for now, leptin. Leptin is a hormone produced by adipose tissue, so it's produced in fat. And it signals to the brain appetite control. So the receptor for leptin is found in the hypothalamus, or region of the brain. And if the body can detect leptin, if the leptin receptor binds to leptin, then the body says, okay, we have enough food right now. But if there's not any leptin binding to the leptin receptor, that's a signal to the brain that there's not enough food in the body and we need to eat food. This is a hormone that controls how hungry you are, and as you can imagine, anything scientific that controls how hungry we are is great for research and great for potential weight loss supplements. So some scientists did a study into mice. They said, hey, we've identified the OB gene in mice that controls leptin production. And if we get rid of the OB gene, then the mice don't produce leptin. So the question is, why are mice that do have the gene obese? Why is it that mice that are OB/OB obese? So the hypothesis was, well, if the mice aren't producing leptin, that stimulates their appetite and leads to overeating. So we tested it, or the scientists tested it by injecting mutant mice with leptin, and the results were, after taking a fat mouse who was obese because of this mutation, and injecting him with exogenous leptin that was made in a factory somewhere, not actually leptin made by the mouse. Injecting the fat mouse with leptin led to the mouse losing 30% of its body weight with no real side effects. An interesting study. Last thing we are going to talk about are essential nutrients. I'm going to define essential nutrients as any nutrients that an animal can't make for itself, but that is necessary for health. So the ones that we care about are vitamins. As it turns out, humans, apes, monkeys, and guinea pigs can't make vitamin C. Some animals can, we can't. Sucks. Amino acids. Humans require 9 essential amino acids. There's 20 amino acids that matter to humans, but 9 of them we can't make through any processes, and so we call them essential amino acids. They must come from our diet. Certain types of long-chain unsaturated fatty acids. These are big fatty molecules that we can't synthesize, and so we need to eat them. And then minerals, ions, metal ions like magnesium, iron, selenium, zinc. We need them for processes in our body, but we can't produce them. We have to consume them in our diet. And then the last slide is a slide full of vitamins that are all very interesting. And all of them are major and essential to humans, and I don't care about any of them because there's a lot of information here. It's nice to know that vitamin A, retinol, D, calciferol, E, tocopherol, and K, vitamin K, a clotting vitamin, A, D, E, and K are water soluble, and the rest are fat soluble. And other than that, if you want to learn more about them, the information's right here. Otherwise, this is all the information that I want you guys to cover for Chapter 34. For the test focused on all the things that I've highlighted and spent time on in this lecture.